Next up, we have Lorenza, and Lorenza Raimondi, whose biography was given to me as this. She did her BSc in biology in Napoli and her master's in Genoa, south and north of Italy. And now, of course, she works in the Cirque lab, and I think in the running for longest title of her seminar. So if you want to get your slides up there, and we can just get going. Also, lots of slides, so it's going to be good. So I'll let you take it away,
There's
Lorenza.
many extra slides.
Okay, good.
Yeah, I actually wanted to apologize because I didn't realize how long my title was until I put it on a slide. But anyways, today I'm presenting my PhD project, which focuses on the temporal variability of anthropogenic and natural carbon in Labrador Sea. And I do this by using measurements of dissolved inorganic carbon as well as chlorofluorocarbons. So first, I wanted to give kind of a background on why the Labrador Sea is an important region to study the variability of anthropogenic carbon. And probably most of you know that the Labrador Sea has one of the highest integrated column inventories of anthropogenic carbon in the world ocean. And more recently, this paper by Gruber et al. Show that can you see my arrow actually? This region is shown to have one of the highest increase in anthropogenic carbon column inventory between the 1994 and 2007 period as well. So you can really see how important the Labrador Sea is for the sequestration and entrainment in the deep ocean of anthropogenic carbon. And so the second reason why the Labrador Sea is an interesting area to study the anthropogenic carbon invasion is also the availability of the long-term monitoring program maintained by the Bedford Institute of Oceanography. This is the Atlantic Zone Offshore Monitoring Program, or AZOMP, which I'm sure a lot of you are familiar with. And here we have annual occupation of the AR7W line that you can see in the map here that crosses the Labrador Sea, where we have measurements of hydrographic data, alkalinity, nutrients, but we also have dissolved inorganic carbon, which is the sum of all the inorganic species of carbon, as well as CFC12, which is one of the chlorofluorocarbons, and these are gases that have been produced by anthropogenic activity as well. And so the Labrador Sea monitoring program actually gives the opportunity to look at the carbon cycle from different perspectives. We can look at the total increase of just the dissolved inorganic carbon, but we can also estimate the anthropogenic carbon by using all this large suite of measurements that we have. And so the first thing we did was to look at the just overall increase of dissolved inorganic carbon. Here I'm showing the average full column GIC concentration in central Labrador Sea. And you'll see that the average concentration is increasing between 1992 and 2016 and at a pace of 0.6 micromole per kilogram per year, which results in an increase of 15 micromole per kilogram over a 25 years period. And this really means that the Labrador Sea has seen a very large invasion of probably of anthropogenic carbon. So the main question that came up out of this plot is that we wanted to know if this increase in DIC is solely due to the anthropogenic carbon invasion, or if there is some natural variability that is capturing this signal but that we haven't characterized. And so to do this, of course, I first had to estimate anthropogenic carbon. And because we cannot really measure anthropogenic carbon concentration unless we have isotopes measurements, we actually use indirect estimates indirect methods to estimate anthropogenic carbon. And there are many out there, but for my PhD thesis, we kind of decided to go with the transit time distribution method, which is a method based on transient tracers. In particular, I use CFC12. And we also use the extended multiple linear regression approach that is a carbon-based method. So we actually use dissolved inorganic carbon measurements for this method, and is specifically designed for repeated hydrographic sections. So it works really, really well for the AR7W line. So I'm going to go a little bit in details about the methods and show you some results from each of them. So the transit time distribution method, as I mentioned a little bit, is a method that tries to estimate anthropogenic carbon by using other gases that have been produced by anthropogenic activity as well. So the idea, well, the method is primarily based on this equation that you see here, where the anthropogenic carbon concentration at a certain location and time is given by the surface history of the anthropogenic carbon and this G function that you see here, and that I'll introduce in a second. So the anthropogenic carbon surface history can be obtained if we assume a constant disequilibrium between air and sea. So if we assume that whatever is in the atmosphere is also in the surface ocean, then we can take the atmospheric concentration of CO2, remove the pre-industrial signal, obtain the anthropogenic increase that we see in the atmosphere, and assume that that's the same that we see in the surface ocean. And then this G function is actually what we call the transit time distribution. So for those that are not familiar with this concept, the transit time is the time that a water parcel takes to go from the outcrop region, so the origin region, to the location that we're looking at. So when we go out at sea and we take a bottle of water as our samples, in that sample, we're going to have numerous water parcels that come from different outcrop regions and had different pathways, and therefore have different transit times. And so we don't have just one transit time in that water, but we have a distribution of transit times. And because we cannot actually measure the anthropogenic carbon, we use other tracers like the CFC12 to assess this transit time distribution. And so we use the same equation, but here the C represents the concentration of CFC12 in my case. The surface history, again, can be obtained by the concentration here is obtained by observations. Sorry, baby crying. From observation, so from our measurements from the Lab Sea. And then the surface history can be obtained, again, by assuming that there is an equilibrium between the atmosphere and the surface ocean. And so we have a constant saturation of the tracers. And because we know the CFC12 concentration in the atmosphere quite well, we can derive the surface history from there. And then the G is our unknown here and can be obtained from this equation and then plug back into the first equation and obtain the anthropogenic carbon. So you see that this method heavily relies on this two assumption of getting the surface history of anthropogenic carbon and CFC by assuming this constant equilibrium in a way between atmosphere and ocean. And this is not exactly true. And a lot of my PhD thesis has focused on validating these two assumptions. And I actually came up with a kind of improved surface history for the tracers. And, but I made this presentation with sea dogs in mind. So I think this part of my PhD is a little bit out of the scope of this presentation. But if you have questions about how I did that, I'm happy to answer them, but I'm not going to go into details here. So the only thing that I wanted to show is the anthropogenic carbon concentrations that we actually got from this method. And I'm going to play a little movie here where on the left side, you'll see it's the Labrador coast. On the right side is the Greenland coast. The y-axis is depth, and light colors are low concentration and dark colors are dark concentrations. And so you'll see as the years go by that the anthropogenic carbon concentration has increased everywhere in the Labrador Sea, but primarily in the first 2,000 meters depth, where we see the Labrador Sea water, where we see deep convection. And as well, you'll see a strong increase at the bottom of the basin, where we have the Denmark Strait overflow water, which is again a younger water mass that has been recently in contact with the atmosphere, and so actually shows a higher signal of anthropogenic carbon at the bottom of the basin. And then we isolated the central portion of this basin, and we calculated column inventories for the central part. And then we look at the time series of these column inventories and kind of saw that there wasn't a steady increase of the column inventory, and therefore we used a change point analysis to look for abrupt changes in this time series. And so we found three characteristic periods. The first one goes from 1986 to 2001. And we see that the column inventory increased at a pace of 2.2 mole per meter square per year, and this is what we call the storage rate. In the second period, we actually see a slowdown in this storage rate to then pick up again with a fast increase in the latest years of our time series. And I didn't show here anything else rather than the column inventory just for shortness, but this behavior of the increase in column inventory, these three distinct periods, actually align quite well with deep water formation times and with the heat content of the water column. And so the second method that we used was the extended multiple linear regression. And this method tries to obtain the anthropogenic carbon signal by removing the natural signal of the DIC. And this is done by exploiting linear relationship that this DIC has with other parameters. And so the idea is that whatever naturally changes the DIC will also change the other parameters. So the linear relationship between DIC and the other parameters won't change over time. And so the method works this way. You have data from different occupations of the AR7W line. So the first year, you derive a multiple linear regression between the dissolved inorganic carbon measurements and some independent variables. And in my case, I can pick and choose because I have a lot of different measurements. Then you go at sea a second time, and you'll have a second multiple linear regression applied to a second time in the time series. And so the idea is that if we take the difference in coefficients from the first multiple linear regression and the second one, and we can use those new regression coefficients in a way to obtain the change in DIC. So the equation is going to look something like this, where we have the difference in coefficients. And so the signal that we obtain is the change in DIC from the linear regressions. And so because I have multiple independent variables to choose from, I actually did many different multiple linear regression with changing both the number and the combination of the variables. I'm not going to show all of them here because it would be a little too busy, but I just want to go in my last slide where I kind of bring everything together. And so here it's kind of how we think we're going to try to isolate the signal of natural versus anthropogenic carbon from the DIC. So here I'm showing the column inventory that I calculated for each year just based on the dissolved inorganic carbon measurements. So you can see these as the total change in DIC as reference to the year 1993. And we saw that this increased at a pace of 2.6 mole per meter square per year. When we then use the anthropogenic carbon estimates based on the TTD, we see that the storage rate is actually lower. And so this would imply that the anthropogenic carbon increase does not fully explain the increase in the total DIC. And so there is some natural variability associated with the signal that we see in DIC in Labrador Sea. then when we use our EMLR estimates of anthropogenic carbon, we actually see kind of a reconciliation between the total signal and the anthropogenic one. And so this would actually show exactly the opposite, so that there is no natural variability associated with DSC. So what I'm working on right now is to actually understand what is driving these differences, and if there is a way we can actually define if there is some natural variability associated with this signal. So to give you some highlights of this presentation, I used a long-term monitoring program dataset to assess the variability of anthropogenic carbon in Labrador Sea. We have two methods to estimate anthropogenic carbon, the TTD and the EMLR. We looked at the temporal variability of the anthropogenic carbon over 30 years and estimated the storage rate for different periods over the time series. And I showed preliminary results on the separation of the anthropogenic and natural signal in the total DIC through both the TTD and the EMLR estimates. So with this, I would like to thank my founding agencies and collaborators, and I'll take any question if you have them.
Okay, great. Again, if you have a question, just use the raise hand function in the participants window. Anyone out there? Nobody.
I have a question, but I can't find the raised hand.
Okay, just go for it.
Sorry. Hey, I'm also doing some sort of statistical learning, so I was just curious how you selected which variables in that second method for the multiple linear regression, which variables to include.
So there isn't actually a way to do that. So it's kind of one of the problems with this extended multiple linear regression method is that it's kind of pretty subjective what parameters you use as independent variables, and most of time you're kind of restricted to whatever measurements you have. So people tend to used to not think about this too much, because you only had, not everybody has the large data set that I have access to, and not everybody has, like yearly measurements. So I think there is more ways to approach this, and one that is coming a little bit more used, I think it's kind of based also on the approach that they use in modeling, where they do an ensemble estimate of anthropogenic carbon. So they would do several EMLR and then take the average of this ensemble EMLR to kind of get rid of a bit of the uncertainty due to picking and choose the parameters.
Oh, thanks. Yeah, I just feel like it would be even harder because you have the multiple time periods. So you would get different importance values for each of the variables for your different regressions. So putting them together, I just sounds very difficult. Thanks for the presentation.
I believe we have a question from Jin Yu.
Yeah, this is it's a really good talk. I quite like it the way you did, and you mentioned that a dynamic model can be used also to estimate the DIC in the Labrador Sea. I just wonder what is missing in your statistical model. For example, we know there are so many other dynamics like winter convection, transport, horizontally mixing. So what are important terms missing in your statistical model? I wanted to think about it, right?
I mean, the statistical model based on the EMLR, it has a lot of flaws, but we get surprisingly pretty good results. The main problem is you cannot really get reliable estimates for surface ocean, because the relationship between DIC and the other parameters is absolutely not linear in the surface ocean. So we kind of have to remove the top, like 200 meters, 300 meters.
Yeah, one of the things is like winter convection, I know is the highly variable each year. And another thing is transport horizontally. So I just wondered, this is a factor being considered in the statistical model now.
Not in the EMLR, but in the TTD, we kind of take into account of that because, so I didn't talk about this, but when I did my validation of the assumptions of the method, so the method assumes that you have equilibrium between the ocean and atmosphere, and this is actually not true because of the deep convection. And so what I tried to do was to model the saturation using, again, a multiple linear regression, but in that case, I used both the atmospheric increase of the tracer and the mixed layer depth for each year. So in that case, we do kind of take into account the extent of the convection and how that would affect the saturation of the CFC, and therefore it directly affects the estimate of anthropogenic carbon.
Great, thanks. Yeah, thanks.